All right. Good afternoon, everybody. Welcome back from the last coffee break, I guess. My name is Jair Siqueira Neto. I'm an associate professor at the Skaggs School of Pharmacy and Pharmaceutical Sciences in San Diego, at the University of California, San Diego. So first and foremost, I would like to express my appreciation and gratitude for the invitation to be here in this very special event celebrating 10 years of LOLA and 20 years of DNDi. So thank you very much, Jadel, Luis Carlos Dias, Luisa, and Artur Cordeiro for making it possible for me to be here today. It's really a great pleasure to be among so many friends and collaborators that all share the same vision and passion to fight neglected tropical diseases. This event is very special for me in two ways. So my involvement with drug discovery and development for infectious disease actually started with DNDi when I was a postdoc at the Institute Pasteur Korea back in 2006. I was there with support from DNDi, and that's when I started working on high-throughput screening assays for Leishmania and T. cruzi. And here I am many years later. And the second thing is that Unicamp is actually home for me. I did my undergrad and PhD here at this university, so it's very special in many ways. So we're now going to dive deeper in antiviral drug discovery. Our first speaker is Dr. Annette van Delft. She's a translational scientist at the Center for Medicine Discovery at the University of Oxford. She works on a range of targets in infectious diseases with project covering target validation, discovering, and preclinical development, with a focus of her current work on the preclinical development of a novel antiviral target in coronaviruses. She also works on flaviviruses and enterovirus as part of the COVID Moonshot and ASAP consortium. So Annette is trained as a medical doctor at the University of Leipzig and received her PhD in clinical medicine at the University of Oxford. So please, Dr. Annette, the stage is yours.
Thank you very much for that kind introduction. And I'm very, very pleased to be here. It's my very first time in South America, and I must say, what have I missed? And it's a great pleasure to be here for such a special occasion. And I just wanted to congratulate all of you that have been in some way involved in all these great initiatives that have been presented over the last 2 days. And we are now going to have a bit of a jump into antivirals, and the next session looks quite interesting to me. And it starts off with a little bit of an experience story, really, and a little bit of a peek out of the box, because what we've been doing over the last 3 years is to do something quite new. We did drug discovery completely in the open, and for us, it was a great learning experience, and I thought I'd just share a little bit about that. So it all started off. So actually, my first interaction with COVID wasn't the science, it was getting COVID myself. And I picked it up before it was officially in the UK, which was in February 2020. And that was around the same time, end of January, this team here at Diamond, Dave Stewart and Martin and Frank were approached by a group in China who had just solved the structure of the SARS-CoV-2 M-Pro, and they wanted to send the constructs over for them to run a fragment screen because they had worked previously together with the Chinese team. And actually this packet with the constructs got lost in the post because of the shutdown that was happening in China. So it took them a while to work out that this packet would never arrive, because everyone was really surprised that there wasn't going to be a quick opening up again and the packet would come through. So they had to clone the constructs again, order them all in. So that was kind of the longest time that it took. But then they very rapidly crystallized the product using the protocols from China and ran a fragment screen. And I know there are a lot of people here that are very familiar with fragment-based work, and we heard that mentioned in the previous talks as well. But what they basically did is they not only looked at the Mpro protein here, but they also looked at all these other proteins and tried to run fragment screens for all of these proteins. So all that data exists, but Mpro was the one that was solved first, and also one of the targets where it was most known what happens with the target. I just put the viral replication cycle here. We don't have to dive into this, but the only thing I wanted to kind of make you aware of is that the viral protease sits here and cleaves the translated polyprotein into smaller parts and is one of the essential targets. And what was known at the start of 2022 was actually quite a lot. So we already knew that the main protease was essential for viral replication. It was druggable. There were chemical probes available that were developed originally for SARS-1. There was some biological target validation for the family of coronaviruses, and there was clinical evidence, not for coronaviruses, but for other viral families, that this would be a worthwhile target to go after. Obviously, this wasn't any new knowledge. There were several pharmaceutical companies that knew that already. But what the team at Diamond really didn't know is how to get from this fragment screen that you can imagine as a lot of Lego blocks to a drug that is orally available, safe, rapidly and readily accessible. And because they didn't quite know how to take that information forward, they jumped on a call with previous collaborators of them and said, so what can we do with this data? Is anyone interested? Can anyone pick this up? And very quickly, actually within 2 days, they had a small group of collaborators together that said, we're keen, we'll do something. Nir Weitzman in Israel said, I'll establish an assay and run that for you. And then there was Alpha, who is at Postera, a small biotech company, who also said, I'm going to help accumulating the data. So the idea of this COVID moonshot, this drug discovery effort completely in the open was born. And what is quite interesting about it is that these first conversations really set the scene for something that was quite unprecedented, because it meant that everyone was going to submit their ideas online, and from the very first moment, all the material would be visible and therefore prior art, so there would never be any IP on any of the molecules. The other thing was that, so this idea of a generic drug straight from the pipeline, the other idea, or the other characteristic of the consortium that came very quickly was, because there was nothing to protect, there were no contracts to be signed. Everyone just chimed in, and that was actually a massive time saving. I remember the faces of the colleagues at Novartis when they wanted to help us with PK, and we said, no, we don't need to sign anything. You literally just order the compounds off the catalog. We pay for the synthesis, and you just get the compounds. And it took them a couple of seconds to just understand that there was literally no paperwork involved. So that was quite a big thing. We also were very lucky to secure a lot of collaborators that work in AI and machine learning, and their experience and their protocols really helped to accelerate the synthesis. And we had a lot of in-kind contributions and crowdsourced funding by everyone just chiming in and bringing their expertise to the project. The other thing that also came is no one from the team had ever met before in person. So everything was completely run on kind of oral agreement and handshake via Zoom. And that created a very flat hierarchy. So anyone could chime in, anyone could come with their ideas, which really created a very unique and also very critical structure, because anyone felt empowered to bring their ideas. So even the PhD students would speak up and contribute, which I found very powerful, actually, as a message overall. So here are some of the people. So some of the things obviously were developed before. We couldn't have started this without any background. The one thing that was established before was this Fragalysis platform, which is a platform where you can basically see data from anywhere in the world and look at the fragment hits in a very easy and accessible way. You can comment on the structures, comment on different fragment parts, and pull your data together in one place and search it easily. So Darren and Frank were the ones who set that up, and that was really the basis for the success. Alpha, whom I've mentioned before, and his team managed to pull up the website to actually submit your structures within 2 days. So within 2 days, we had this website and anyone could use it, could put their synthetic rationale in, it would be visible to anyone. And that was the main tool that the Moonshot project was carried about. And in retrospect, we realized how crucial that was, because it was our central data suppository, and it was also the place where we created the prior art by publishing the assay data to the structures as soon as we had them. And then we were a bit surprised by the amount of interest we got. So within the first week, we had thousands of ideas submitted. And we did say, or we did promise in the very first tweet, that we would make these structures and get them synthesized, but we hadn't really expected that much of a response. So when you have so many ideas to cope with, you have to kind of rank them somehow and decide what you can do. And we had another restriction, which was that we didn't have a lot of money. Everyone just piled in with research funding that they had left over from a corner. And what then helped enormously were these 2 AI tools that we built into our pipeline. One of them is a synthesis-based tool that was developed by Postera, a new biotech, which basically looks through the Enemin real space library and synthetic pathways and predicts compounds that are very easy to make within 1 or 2 or even 3 synthetic steps. So that was one important part. And the other important part was the free energy calculations that John Cadera from MSKCC contributed to the project, which basically tells you what is a predicted good binder. And in this process, he happened to build the biggest supercomputer. And then the MedChem team obviously had to kind of look through these ranked ideas and still sort some out and decide which ones were going to be made first. We had over 18,000 total submissions. 2,400 of those we were able to synthesize as part of the Moonshot project. And you can see here that most contributions came from the US and the UK, maybe representing our reach within the project, but we, especially at the start, had quite a lot of international contributions as well. The design make test cycle was very much influenced by the people that I've pictured over here. And I want to point out Tatiana, who sits at Enemin. Enemin was one of the crucial partners because they picked up all the logistics, which is something that should never be underestimated in a drug discovery project. So they were crucial to the process. And then very quickly, within a couple of months, we had over 40 groups collaborating. And if you imagine that you have to ship samples, different samples to 40 groups, you can kind of get an idea of what Enemin shouldered, not only the synthesis, but also the rest. These were also a lot of other collaborators that helped us with viral synthesis, ADME and PK. And we were then very lucky to get DNDi as a partner on board to progress the compounds into lead optimization, late lead optimization and early preclinical development. And that work has been very kindly funded by the Wellcome Trust. Some of the learnings that we had. One is, if your data is available, people will use it, especially if it's a data set that links structures to biochemical assays. You can see here the different data releases. You can see that these are the different snapshots that were taken on Fragalysis. And we worked back to find out that actually this provides an excellent medicinal chemistry teaching tool. So I encourage all of you, if you can, put your data out. Other people will benefit from it if you don't use it anymore or you don't have to IP it. The other thing we learned is that Shionogi, who now have a clinical inhibitor in phase 3 trials or approved in some regions, has used the moonshot data to inform their pharmacophore in the early stages, and we count that as a massive success for the moonshot. And the data has been extensively discussed anyway. So the preprint has been discussed. We're very excited for a science paper to come out soon, so look out for that if you want to see more details on the data. One of the other things I wanted to kind of just put a quick light on is the ASAP consortium. Some people of you may have heard about this. This is one of the NIH-funded centers. It comprises the Moonshot team. And the aim is to look for antiviral inhibitors for 3 different families: coronaviruses, flaviviruses, and enteroviruses. If you can read the names, you might recognize some of the names again. Some of the partners that have been working with us previously, and DNDi is involved in the preclinical development of the compounds. What I would like to point you to, and you can explore that in your own time, is the website that not only details the data, but also independently verified and verified structures, plasmids, all the protocols that we have been using. This is an example of the deep mutational scanning data that was derived by Jesse Bloom in Seattle. That is all openly available. You can click, this is an interactive website, you can click on each of these amino acids and find out which are the conserved sites that you might want to target in your own projects. It also includes what we call target enabling packages, so expression data, purification processes, assays, the X-ray structures, and early hit compounds. We still have a glitch on the early hit compounds, but they will be online soon. So please use our data. That's, I think, the main message. It's hidden away in the corner here. We did learn quite a lot on the way, and DNDi actually has been quite crucial to help us work through the process and think about equitable access and availability of the compounds. The thing that we have learned through this crowdsourcing process, and I appreciate this might be a project that may not be replicated in the same way, is early discovery, if it's done in this open access way, is comparably cheap and is very easy to do. But if you do it, licensing for access needs to be thought about from the start. And we also do think that this might be an idea to look into for pandemic preparedness. We also had quite a lot of challenges on the way. One of them was logistics. We had 3 wars so far that impacted the project. And at this point, I just want to kind of have a shout out to our Israeli and Palestinian colleagues that are still in the lab and still measuring our compounds, which I find absolutely astonishing. Funding is a key point, and this has been brought up before. Open science is sometimes difficult to fund because the setup is often not there yet, and you have to kind of sell it as a worthwhile thing to take forward to a funder. And finally, I want to thank everyone who has been involved. Please get in touch if you want to work on some of our proteins or use our data. It's all there for you.
Very nice talk. I love hearing about backstage stories in science, and very impressive how fast you act. So our next speaker is Professor Vivian Vasconcelos Costa. She's an associate professor at the Department of Morphology at the Institute of Biological Science at Federal University of Minas Gerais. She won in 2020 the For Women in Science Award promoted by L'Oréal UNESCO in the Brazilian Academy of Science. She's also a member of the Technological Center for Advanced and Innovative Therapies, CT Therapies, at the Federal University of Minas Gerais. And she has experience in the areas of microbiology, cellular biology, and immunology, with an emphasis on cellular immunology, virology, and microorganism host interaction, working mainly on the following topics: immunopathology of infection by dengue, chikungunya, myrovirus, yellow fever, Zika, and coronaviruses. She has also interest in viral and autoimmune arthritis, with emphasis on the development of experimental models, including humanized models, to study pathogenesis and test therapies. Vivian.
So I would like to thank the kind invitation. I'm here today also on behalf of Mauro Teixeira, which we share the labs, and he's a clinician. So nowadays I'm doing a lot of the preclinical work, and we're trying to put together the clinical work. So this is the interest of our lab. We study a lot of arboviruses, so today I will focus on dengue, but we also work with yellow fever, Zika, chikungunya, Mayaro, and most recently we are working a lot with coronavirus infection. We also have models of influenza, monkeypox, and other infections ongoing. So in the past few years, what we are doing is trying to set up preclinical platforms to study these viral infections disease. So we have different mouse models available to test all the compounds that we must be interesting. As I said, we have, for example, Zika models of acute infection, but also congenital infection and others, so on. So, but what does disease has in common? It's the viral factors can contribute a lot. So we try to figure it out, these viral factors, the proteins, the virulence factors, which are related with the severity or the of the disease. But also, what we do a lot is to study the host immune responses unleashed by those infections. So we know that an excessive or misplaced inflammation, most of the times it's really important to lead the severity of this disease. But more recently, 20 years the past 20 years, there are some groups, including ours, which are hypothesizing that this disease may be, the severity of this disease, maybe it's because of a failed resolution of inflammation, and I would like to talk a little bit more. So our work hypothesis in the lab is that the prevention of certain aspects of the inflammation maybe could be a good strategy to control the infection as an anti-viral strategy. I mean, if we can target the virus, we can prevent the disease, or also if we can prevent some aspects of the inflammatory response as an anti-disease strategy. So today, I would like to share some data that we are working in the past 6 years, in which we asked if dengue is associated with an altered resolution of inflammation. And if yes, maybe this contributes to the severity of the disease, to the pathogenesis. And if we can intervene in this phase of the inflammation, we can ameliorate the disease. So as I said, a lot of people in the field of dengue, it's putting a lot of efforts in studying the acute phase of the inflammation. So in the phase which we have the viral and the inflammation as well, a lot of mechanisms of severity here were described as well, mechanisms associated with the protection. But what we are doing now, we are very interested You understand this phase of the response, which is the phase of the resolution inflammation, because the inflammation is necessary to clear the pathogen. So we have the insult here, and then we inflame. We recruit a lot of cell types, which will release inflammatory mediators and also clear the pathogen. Then we can resolve of the infection. But this phase here, it's really important and it's really active. It's not passive as a lot of people use it to think, and depends on the release of different inflammatory mediators in that environment to shift the recruitment of leukocytes and clear the infection. So resolution, it's an active process, and it's driven by chemical signals, which are different from those ones in the beginning of the infection. So I put this slide here. We worked a lot with molecules to try to reduce this inflammation. And in this context, we can use antiviral drugs and also anti-inflammatory drugs to try to reduce a little bit the intensity of the inflammation. However, right now we are trying to do, it's using pro-resolving molecules, which will act in this phase to try to shift this curve and resolve faster the disease. And there are a lot of pro-resolving molecules. Most of them are tested a lot in sterile models of infections, so arthritis models and no infections models at all. And we are trying to elucidate the role of some of these molecules. Today I will focus on this protein, which calls annexin A1. So annexin A1, it's a 37 kilodalton protein, which binds in a GPCR receptor called FPR2, and then mediates a lot of anti-inflammatory and also pro-resolving molecules. So basically, what annexin does, it's shift this microenvironment, try to eliminate the excessive neutrophil infiltrate and burst the macrophage to clear the environment as faster. So for that, we have some platforms to study dengue disease in our lab. The first one, we use immunocompetent mice. This model is kind of dengue fever model, which is self-limited because white-type mice are very resistant to dengue virus infection. But we inoculate these mice and we look up to 96 hours, the disease is resolved in them. So what we generally see, it's a drop on the platelets, which it's called thrombocytopenia, which it's a major hallmark of dengue virus infection. Increase of hematocrit levels, which it's accompanied by the Evans blue extravasation in some organs. So resemble the vascular leakage that we see in the patients, and also the production of some cytokines and chemokines. This mediator here, it's really important because it's exclusively released by mastocytes, which it's associated with the plasma leakage. It's an analog of the kinase in humans. So then we asked if the annexin levels are changed during the infection, and also the expression of the FPR2, the receptor of annexin. And what we saw in the mice, it's after infection, we have an important drop of the annexin A1 levels in these mice. And as a compensatory mechanisms, increased the expression of the FPR2 in the liver of these mice. Then, okay, annexin looks like to be important, but there is a functional relevance. So we used knockout mice for those molecules. And what we saw is in the absence of annexin A1, the disease it's more severe, than in wild-type mice. And even, which is more important, takes longer to resolve in comparison to wild-type mice. So looks like that annexin, it's a key protein mediating the resolution of inflammation in the context of dengue. And this also occurs, it's similar results were found in the mice, which doesn't have the receptor of annexin-1, suggesting that annexin is acting on this receptor. Then, what we did is to synthesize a peptide, a mimetic peptide, a small portion that binds in the FPAR2 and mediates the function, and give it back to the wild-type mice to see if we can see an amelioration. And yes, we saw amelioration of the platelet levels and also no vascular leakage and reduction of the inflammatory mediators after the treatment with the peptide. And just to comprove, which it's mediated by FPR2, there is no effect at all when we do the treatment back when in the FPR2 mice. So finally we asked, but we need to control the inflammation, but we need to take care that the viral titers is not going up. So how about the viral loads? This model, it's not good to check the viral loads because it's a self-limited model. Then we went to another model, which is the gold standard for dengue. It's the A129 mice. It's type 1 receptor interferon deficient. And we did the treatment with the peptide as a therapeutic scheme starting at day 2 of infection. Then what we saw, it's the peptide prevents a little bit the weight loss, also the thrombocytopenia induced by the infection, and really nice results without change the viral titers. So we are changing the inflammatory response, but not the viral titers. Then we went to humans to confirm if this is true for humans. We measure annexin-1 levels in patients with dengue, and we saw a reduction of annexin-1 in these patients. And if we segregate these populations in patients with mild dengue or severe dengue, we do see more drop in the annexin-1 levels in the severe patients, suggesting that if this protein is important in the severity of the disease. So we can argue here that the use of the pro-resolving molecules maybe is useful as a coadjuvant therapy for dengue. So then just a few results to finalize. We are testing some antiviral compounds from different places. This is a nucleoside analog from the Emory University. We are working with some compounds for a quite long time. And today I will show this same compound that we just published for yellow fever for dengue in the same model of A129 mice. And what we see is the antiviral compound also prevents the thrombocytopenia and reduces partially the viral titers in all the places that we look at, even the brain, because the viral goes to the brain of these mice as well, without change any inflammatory parameters in these mice. So we have cytokines, but the compound does not reduces them. So then we are starting to do, this is a very, very fresh experiment association. So what we do see here is that the vehicle, it's here, this is a clinical score, and all the mice succumbed to infection by day 8. And this is just the antiviral, which we started really early, 1 hour post-infection. And that's no difference in the clinical score at all. but the peptide, the anti-inflammatory peptide, ameliorates the disease, but the combination does not reduce in the prevention of lethality. But we do believe it's just an arrangement in the therapeutic window here. We will play with that. So I just would like to thank all the funding, the opportunity to be here to share our work, and it's a lot of people that work in these studies, and so I would like to thank. Okay, it's okay.
Thank you, Vivian. Great talk. So the next one is Tiago Moreno. He's a full professor at Fiocruz, Brazil. He's a virologist and biochemist with experience in coordinating studies in the area of repositioning and development of antiviral viral diagnosis and sequencing and virus cells interactions. He's the Deputy Coordinator of Innovation at the Center for Technological Development in Health, CDTS Fiocruz, and his group participates in very collaborative initiatives such as the Metropolitan Antiviral Accelerator, MAVDA, and the AVID Center as well. Tiago, welcome.
Thanks for the invitation. I'm Tiago Moreno. I'm going to present to you this from HMB 905, which is a partnership between Fiocruz, which is belongs to the public sector, CIEMP from Callisto, that you all know. It's a private, non-profitable organization, microbiologica, an active ingredient producing factory, which is a profitable company. So this was the day when our life changes in this country. Policymakers and many people tend to lead to the notion that the worst is gone because we have lower mortality now, but people still die from COVID-19. And taking Brazil, for example, cases of acute severe respiratory infections still occur in 2023 in all ages, but especially the elder are those that die. So this is one reflection. Should it be considered for a target product profile, for example? COVID-associated death relies on the effect of the virus to kill type 2 pneumocytes, the surfactant-producing cells, and also produce apoptosis in type 1 pneumocytes, lead to cellular infiltration, pro-inflammatory cytokine storm. Intense cell death occurs, as well as tissue disruption, leading to coagulopathy. So we know that from pre-symptomatic to critically ill, some treatments had been proposed, mainly to the late stages of disease. Some antiviral were approved, especially Paxlovid. Some caveat exists for molnupiravir and remdesivir. But Paxlovid has many drug interactions, and this could be critical for the population that is most dying. But we wonder if some antiviral could reduce the virus-induced inflammation, if we could have a wider time frame of opportunity for treatment. And our interest relies on the virus replication machinery, which is quite different from this virus. This virus is able to transcribe the virus RNA into subgenomic and also replicate the virus genome using a polymerase, which is proofread, and which the activity is proofread by an exonuclease. And the story I'm going to tell you, it's published in this article by us, which represents the development of kinetin, the MB905, which is also a natural product. So our primary assay relies on use hepatocytes or CALO3 cells, which mimics type 2 pneumocytes at our hands, and in fact then recover the supernatant and titer to measure the infectious virus titers. And we can see here MB905, which is an adenine analog. It's equivalent nucleoside, and its nucleotide monophosphate as a peptide. They all could inhibit in a dose-dependent manner SARS-CoV-2 replication. Panel A, we just had the drug once. Panel B, the drug was re-added 24 hours. And the compounds were better than molnupiravir and worse than remdesivir. But we want something that could also prevent the virus-induced inflammation. So in fact, human primary monocytes measure the virus, the cell-associated viral RNA. We don't have productive infection in these cells, and the cytokine levels. Here we have the inhibition of virus replication. All our compounds were as good as remdesivir to reduce cell-associated viral RNA. They could reduce IL-6 levels, and especially MB905 could reduce viral-induced TNF levels, which is important to us. And we kind of cherry-picked the kinetin among other compounds we tested, because there had been described a previous use of kinetin in patients with this rare genetic disease called familial dysautonomia. And these works, they highlight this pharmacokinetics for this compound. And we saw that if we simply plot our in vitro pharmacological parameters within that pharmacokinetic for the patients with the rare disease, we would have plasma exposures within the range of activity. So we prioritized this compound, and we thought that could be easier to push it forward in Anvisa and other regulatory agencies. But we had to make sure the drug reached the lung, and indeed it does. As we can see here, when we treat the mice orally, we saw the drug in the plasma for 24 hours, and also in the lung, again, reaching concentrations beyond the in vitro pharmacological parameters. We analyzed the metabolites, and per oral treatment, we know that MB905 is uptaken by salvage pathway to the monophosphate, and the triphosphate is produced in the liver. 5' nucleotidase release the nucleosides in the blood, and the nucleoside is rephosphorylated again in the lung. And this is pretty much the same activation pathway for favipiravir. The mechanism of action, since it's the triphosphate, we assay the triphosphate against the viral polymerase and compare it with the triphosphate for remdesivir. Again, remdesivir was more potent, but our compound also inhibits in a dose-dependent manner the polymerase activity. Also, we measure in CALO3 cells the cell-associated viral RNA, the genomic and the subgenomic. This is the replication, this is the transcription. We saw a more pronounced effect on the replication than on transcription. We tried to see if we could pick resistant mutants, so we grow the virus in the presence of the compound, increasing concentrations of the compounds as well. We grow the virus in the absence of any drug. And in fact, what we saw was two very different viral populations grow in the absence and the presence of the virus. And there was no specific mutations in terms of amino acids. In fact, there were an increased error-prone replication with T2C, C2G mutations. So we decided to take a different approach by treating the virus with the C90 and making several passages with a high effective dose and compare with the virus growing in the absence of treatment. So here we have in panel A MB905, in panel B molnupiravir. This is the first passage, the second, the third, and the fourth passage. We can see mutations accumulating during passages, which is indicative of a rare-prone viral replication. It's mutagenic for the virus, but it's not mutagenic for the host because it was negative for Ames test. This is a test molnupiravir failed to be negative at. And the polymerase activity is also error-prone, but it is corrected by exonuclease. And there were workings suggesting that the HIV integrase inhibitors could inhibit the exonuclease. And also our work from my group with Jing Zhu from Columbia University, showing that NS5A inhibitors from hepatitis C could also inhibit the exonuclease. And we assayed all these drugs. I show here dolutegravir, which has the most favorable pharmacokinetics profile among these drugs. As you can see here, the titers of virus replication, dolutegravir alone doesn't make any effect. MB905 at 10 micromolar reduces 1 log. MB905 plus dolutegravir 2 log inhibition, showing the synergy. The same happens with the nucleosides, 801, the nucleotides, 711, and even with commercial drugs like sofosbuvir and tenofovir. So it's quite consistent mechanism. We titrate the full dose of dolutegravir and MB905. The red area indicates synergy, showing that inhibiting the polymerase and the exonuclease is a double hit. For the in vivo efficacy, we use K18 mice, and treatment starts 1 day after infection. We want to mimic the treatment. We use a very lethal challenge. MB905 could protect some animals, reduce the viral load, reduce IL-6 levels, TNF, and cell damage as measure of LDH. The histology with, for the infected untreated mice, a lot of hemorrhage and double standard viral RNA, which are reduced, the double standard viral RNA, and the lung histology is healthier in the infected and treated mice. We jump it to hamsters. Hamsters have a faster metabolism, so I had to treat with MB905 twice a day to have a significant result. And again, histology was improved. Let me show you. Here, 3 days after infection, the lung of the infected hamster, infected and treated. 7 days after infection, untreated hamster. So there's a good protection. So our history is that from adenine, we studied MB905, and now we have the first pilot studies from the pills with this compound. This was a partnership, as I said, between Fiocruz, Microbiologica, and CIEMP. We are proud to verticalize the discovery, synthesis, optimization, scale up, antiviral, and no clinical characterization in Brazil. This was done during the time frame of the pandemics. MB905 was cherry-picked from other compounds with submicromolar potency. We prioritize it because of the prior human use. It reduces the virus replication and the virus-induced inflammatory cytokine release. It inhibits the virus RNA synthesis in error-prone mechanism. It showed good oral absorption and non-clinical characteristics to push it forward. It decreased in mice replication, lung necrosis, hemorrhage, and inflammation in vivo. So we submitted all these dossiers along with a proposal for clinical trials to Anvisa. Discussions are ongoing. But since the theme of this conference is to advance neglected disease drug discovery in endemic areas, we thought it was important to highlight the contributions in this ecosystem of innovation we had here. So our role was related to the antiviral discovery and to the mechanism of action. To do the in vivo assays was quite challenging because of animal facilities at the level 3. In Rio, our group used an area presented at the Cancer Institute. And we also made partnership with Vivian to study the hamsters at Minas Gerais University. Microbiologica is a different company. It's a science-based company, and they produce active pharmaceutical ingredients. They exist for a long time, and they contributed to the Brazilian HIV/AIDS program. They did the first generic of AZT before there was a law for generic drugs in Brazil. And they work with Pharmasat in the sofosbuvir development. They were in our network when the Zika outbreak started to occur in Brazil, and we demonstrate that Zika, chikungunya, yellow fever are susceptible to sofosbuvir. And one of the bottlenecks is to go from the active ingredient to a pharmaceutical formulation, and they connected with FUNAD to do a pilot study of these first pills that are produced. And CIEMP is well-recognized center in Brazil for non-clinical research on safety and scale and pharmacokinetics. So whenever I found something interesting, there were some modifications, and immediately we evaluated the PK profile. So the capacity to scale up here, the first grams and then the first kilo of the compound was very important to make stability tests and to identify associated substances. All I tested is 99% pure. We connected with Anvisa, and the plans are to do phase 1 clinical trial with this CRO here in Brazil at Goiás State, and here in São Paulo State, maybe do the phase 2 clinical trial with HCOR. Replication is innovation in our perspective. We have other products in the pipeline on this collaboration that are going forward. Two other compounds for COVID-19 and another one for dengue. The catalyzer is the funding. So CIEMP got the first funding for this research through connections with the Minister of Science and Technology and a demand through our governmental agents. There is this program in Brazil for companies to diminish their risk by investing in science through putting their money together with money from Minister of Science and Technology. It's called EMBRAPRI, this program. From state agents of Rio de Janeiro, I was also able to raise funds. And now we are trying with Fiocruz additional funding for the clinical phase and formulations. So this is what I had to show to you. I'll be glad to take any questions afterwards. Thank you again.
Thank you, Thiago. So last but not least, we have Dr. Daniela Trivela. She's the head of drug discovery of LN Bio at CNPen here in Campinas. So she did her bachelor's degree and master's in bachelor's degree in biology and master's in biotechnology at Federal University of Santa Catarina. And then she did a PhD in science at University of São Paulo. And she did a postdoc in 3 different places. So here at Unicamp, she did also at the University of California in San Diego, and at the University of Nottingham, with additional training in project management at UCSD. So her main scientific interests are on discovery of novel bioactive natural products, proteins, allosteric modulators, and their development into new drugs. She develops also new tools for discovery, characterization, and production of new natural products, and customized derivatives for drug discovery projects with academic and industry. Daniela, welcome.
Thank you. Thank you, Jair, for the kind introduction, Jadel, Luis, Luisa, and all the LOLA 10 organizers for these wonderful 2 days here with you. It's been really amazing. Well, I hope I can deliver my talk. I will speak of the drug discovery platform at LNBio, especially the innovative technologies for natural product drug discovery. We are located at Senipen, which is like 5 minutes from here, and in Campinas. We are at the Brazilian Bioscience National Laboratory, which is just here, one of the national laboratories from Senipen. And we work there in a very interdisciplinary environment with physics, biologists, medical doctors, and everybody who wants to contribute with the science and innovation in the campus. Well, we have the very intense and interesting section this morning regarding natural products, so I think I don't have to explain to you how they are a very important source for new drugs. The problem is that we have many bottlenecks, and one of them is in the very early discovery, to compare it to synthetic approach in which you screen and you know immediately which molecule is there, is your hit. For novel natural products, you generally screen crude extracts or fractions, searching for new molecules, and then you have a long way to find the molecules, and especially if they are real binders of your protein, real ligand, and even more the structure of these molecules, and it can take a long time. So one thing that I focus in this talk is how to do this faster and have as soon as possible the information of the natural product, bioactive natural product, chemical structure, and its binding site in the target protein. So with this, we can quickly obtain this information and feed modern drug discovery platforms, and then use the information from natural products as inspiration for developing new drugs. So how you do that? I don't know how the place is better. So our strategy was to join Brazilian biodiversity with a very good competitive differential that you have here in Brazil, with many molecules still to be discovered, with 21st century technology available at the Senipen campus, including the 4th generation synchrotron light source, high throughput screening, high throughput crystallography approach, metabolomics, and high performance in computing. Because you can generate lots of data, so you have to be able to mine this data and obtain the information that you want. So for doing that, first we establish a drug discovery platform at the center. So I think you are most of you are very familiar with this logic, in which we start with chemical libraries, targets and bioassays, find hits, characterize them, optimize the hits into leads, and then test for proof of efficacy, improving the molecules to obtain the candidates. But to work with natural products, we have to, oops, my life insurance is not, we have to develop new tools and softwares to be able to start with innovative chemical libraries of natural products, Brazilian natural products, and then use these methodologies and softwares to be able to obtain the hits as soon as possible in the pipeline, right? So that's what I will talk to you today. So basically, the challenge is to start with prefractionated natural product libraries from new botanical or microorganism samples, and very quickly obtain the structure of the bioactive natural product bound to the target protein. So then we have the system that you want to apply in structure-based drug design. So what do you do to achieve that? So first, our chemical libraries are very systematically prepared, in which we have the crude extract here in black, and 9 fractions derived from this crude extract. So it's a very systematic way of preparing that and organizing the sample into 384 well plates, in which we're not thinking now in isolated samples. We are thinking in chromatographic series. So each half-life of the plate have 1 crude extract and 9 fractions, 1 crude extract and 9 fractions, and so on. So systematically organize it, because then for data mining, you need this information, right? So this library is very compatible with high-throughput screening methods, so you can select a target, a protein, or an enzyme to find inhibitors, for example. You screen, and then you find a hit, like a bioactive fraction, and then you look at this fraction in the context of its chromatographic series. So you have a crude extract, less active, and a fraction that's more active, and some fractions that are moderated, active or inactive. So this is very important, because the next step is to compare the bioactivity values of the different samples and the presence of the different compounds in these samples. So all the library is already scanned by mass spectrometry. Jean have talked about that technique earlier today. So basically you have the MS2 spectra of virtually all the compounds present in the unknown samples there in the beginning. So you have the barcodes, let's say, of the samples. And as you are working in chromatographic series, you can relatively quantify each molecule in the different samples, and then you can apply bioactivity correlation coefficient, which is to compare each spectra, the distribution across the chromatographic series versus the bioactivity data. So to each spectra you have detected in the samples, and say comparing hundreds or thousands of spectra at once per chromatographic series, we can extract the bioactivity correlation coefficient, which is a score. So you can rank all the molecules and see the ones that have the higher score. That is the most probable natural product or spectra in this moment to be the bioactive compound. Okay, so then you have a candidate spectra, but how can I get to the molecule? That's what I want, right? Nowadays we have many spectra databases, even for experimental or in silico data from natural compounds. So you can compare our spectra with this database and then annotate chemical structures. So this is very useful, because if the molecules are already known, you have a very good score in structural notation. Or even if it's similar to a known compound, you have an indication which structure is there. You can also analyze these in your set of heat through molecular networks that Jean also introduced to you today, and see if you have who is your bioactive compound, if you have analogs, if they are present in other heats, or even you have analogs for further design. So this seems very complicated, but we have developed a software, the NPQ BMS workflow, which is an open source software, and it can take the raw mass spec data and the raw bioactivity data at once, and then do all the processing and biocorrelations automatically. So it's very fast. Then you can have your candidates and structures annotated. If you are working on a target-based project, one of the criteria that you use to rank the projects to enter in the pipeline is if you have crystal structures of these targets, because then you can use protein crystallography as a tool to directly capture the bioactive compound from the complex mixture there of the library. So basically what you do, you take the bioactive sample, full of compounds that you don't know yet which they are, only for the mass spec data. You incubate with the crystal, and the crystal itself will fish, I say, the bioactive compound into the bind site in the protein. So this is very important because you know that there is a compound binding to the protein, because that's a problem in natural products. Sometimes you have bioactive compounds, but they're not real ligands. You know which binding sites they are, and you have the shape of the molecule. So then you can compare the electron densities with the suggestions from mass spec, with two orthogonal techniques having more sure about the molecule that you have there. So here is one example with a very a protein. It's very familiar, I think, for most of you, the proteasome. It's a huge complex. The crystals are not the easiest ones because it's an isotropic, moderated resolution. And here is one example of a fungi natural product captured into its bioactive site in the proteasome. So this electron density that you can find there, you can then start to interpret it with atoms, deduce the molecule, and also fit and compare with the mass spec data. So here you have a molecule bound, which corroborates with the experimental crystallographic data. You have the binding sites, the atomic interactions, and so you have a system that you need for applying structure-based drug design approaches. So this is very sensitive, as we could capture the compound, even from the crude extract in which we couldn't detect the molecule by mass spec. If you use the sample from the library, you have a very more clear electron density. You can start to detect the molecule by mass spec. So the importance is to use prefractionated libraries. And even if you isolate the compound, you don't have a very good incrementing the information that you have. What you have from the fractionated library is enough to interpret the molecule, right? So the point that we start to do that with many different proteins, different enzymes from different classes, and we were successful in most of the projects. We're having many binding sites and electron densities to interpret. So it's become very time consuming to interpret all the electron densities, right? So what we have done was, and I have the pleasure to introduce to you, is the League PCDB database and the NPQ blob label application. The League PCDB database is a database of ligands, all ligands from the protein data bank you have extracted, and use it point cloud to represent the electron density, which is a format very compatible with deep learning architectures. So you apply some filters and tested different chemical vocabularies to be able to teach deep learning architectures to automatically interpret the ligands, even unknown molecules. And then you transform this database into an application, which is a software that will be, is also open source, will be soon available to all the community and even the database for further deep learning applications. Basically, this application, it extracts the blob of the ligand, which is the residual electron density relative to the capture molecule, and then it applies semantic segmentation of this image, giving the substructures of the ligands. For example, cycles of 6 or 5 atoms, and then the types of atoms, oxygen, nitrogen, carbon, and then you can build your ligand semi-automatically. So very straightforward and very useful, especially for new molecules. And it's important to mention that there is no currently available deep learning tools for automatic segmentation of ligands from crystallography, and these will be shared with the community for use and improvements also. So, and these applications had a very good accuracy, which is good for us also, and complemented our pipeline for new discoveries from natural products. So with these, we have applied then mass spec and crystallography and these new softwares to then make the natural product drug discovery faster, hit discovery faster. So you were not, of course, the same timing as synthetic libraries, but I think you achieved the optimum timing to obtain these images. And with this, we are doing many projects at the moment with different companies. Ache was the first project that we start. Nintex, Crystalia, and most of them started later last year, and they have already completed this first phase and are progressing in the pipeline. And most of these projects are also funded by Embrapa, which is a very important option that you have now in Brazil to mitigate the risks and do radical innovation in the country. Just to keep in the context of our antiviral session, this platform is open for all types of therapeutic areas. You have also antiviral projects running on, but it's open to everything, especially if we have a target and a crystal, so it can use the full platform. But just to keep in context, and I would like to just share with you 2 slides about the infrastructure for antiviral research at CNPq. Now we have an infectious disease group, a virology group. We are doing the drug discovery, especially target-based. Once you find molecules, then you have to test them in the virus. So you just built a BSL-3 laboratory that is under commission. And this will be internationally certified. Will be the first one in Brazil. It's almost done. It had already started. Some of you have the opportunity to visit a few days ago. The lab is just there. It's already on. And soon you'll be able to start doing the experiments. This lab will be able to do in vitro and in vivo tests. And just to finish, there is another project now at CNPq, which a big challenge is to have a BSL-4 in Latin America. The project is named Orion, which is also a constellation near to Sirius, and this lab will be built just across Sirius, making it possible to do viral research in BSL-3 and 4, and also to have 3 beam lines that go to the BSL-3 and 4 labs. So it will be able to do experiments with mouse, cells, and even to see intracellular compartments with this new infrastructure. So it will be soon available for all the community in the world. Now it will be the first type of lab in the world, coupled to a synchrotron. It's open for the whole community, okay? So with this, I would like to thank all the collaborators, this wonderful team, which is the main team for drug discovery. We have partners here at Unicamp, companies, and natural product groups, and the whole CNPq is working together to achieve these goals. And I also like to invite you, like this photo was taken last June, in which we did CNPq's Open Science Day, that were 16,000 visitors. And next year you'll do again. So if you are here around Campinas, please come and visit us and all the installations and everything. And thank you all for your attention.
All right. I would like to invite the speakers to take a seat on the table now. We open the session for questions. I can start the question. So Thiago, very exciting work that you did with the MB905. So 2 questions. What is the timeline for Anvisa to review the package of data that you sent to start first in human safety trials? And the other one, have you compared activity of your molecule with remdesivir or molnupiravir that are acting in the same mechanism of action and currently approved?
So I'll take the second question first because it's easier. We didn't compare in every step. So remdesivir is more potent, right? But when it comes to pharmacokinetics, our compound has advantages over remdesivir, especially to reach the respiratory tract and by the fact of its oral disponibility. So, and compared to molnupiravir is positive for Ames test. It's a test that evaluates its potential harmful effects on the genetic of the host. And we were, MB905 was negative for Ames genotoxicity, everything related to mutagenic to the host. There's another issue that molnupiravir was very toxic for myeloid cells. So when we did experiments with monocytes, we couldn't reach 10 micromolar with molnupiravir. So this is a concern. I understand molnupiravir is still authorized, but not approved. So, and so I think there is still an open field here. With respect to Anvisa, they kind of have, I don't remember if it's 30 or 60 days after we send the package, the first package. Thing is that they don't evaluate the preclinical, they evaluate the complete scenario. So you request to Anvisa a clinical authorization for a clinical study with registration of a product. And by doing so, you have to support this claim by providing all the preclinical work. And it may go back and forth many times. Right. For example, after the first round of discussion, they request us to do experiments with 40% of fetal bovine serum in the culture because they were concerned of some plasma binding of our compound. We did, they were happy with all the experiments, and now they moved to another phase where they want to see the analytical data from pharmacological formulation and stability, and these things take time on their own, so there's no way to rush it. The thing that gets more complicated is that as the pandemic changes and cases change and become more seasonal, we have to rethink the phase 2, 3, and perhaps the phase 1. So I think that's the most challenging thing throughout the process, and assure the resources to go it forward.
Very nice. I'll keep my fingers crossed.
So are mine.
Yeah, question here.
Peter Sjö, DNDi. I have a question. Really great talks, everyone. But to Danielle, I'm absolutely fascinated by the things you're doing. And I'm just wondering about the kind of automated fitting of structures to the electron density. Is there any kind of restrictions to the size of the ligand? So can you do big peptides, whatever, and fit that to the density in the same way?
Good point. In this case, no, because we use fragments. So we divided the ligand structure in substructures, like the simplest that you could. So then you can mount any type of a ligand. And that's interesting because there are some solutions now for automatic interpretation, but based on fixed molecules. No? So if you have new molecules or things with size and so on, it's complicated to interpret. So we said, let's divide in small pieces, and then you can then mount the puzzle. No? The machine does that for us. So that's the idea. So the basic structures are atoms, like the carbon, oxygen, nitrogen, the common ones, and cycles. That's it, the restrictions.
A question there in the back.
Not working.
That microphone is not working. It's the same one I used, I think. I've been tricked with that one too.
Sorry for that, I turned off. Daniela, have you ever compared the efficacy of your software with standard approaches, like for known ligands? Because you have a unique approach by subdividing. Have you ever compared how much, because this is a pain in crystallography, you know that those softwares to build
Yes.
ligands are not good.
Yes, we compared the solutions available with the accuracy. Everyone uses a different metric, so it's quite difficult to compare. But the one available has 200 ligands, and most common in protein crystallography, and have the same or even lower accuracy than we do with the deep learning approach. So it's interesting. Yeah, so I think that's the
I'm sure Frank will want to talk with you about that.
Yeah, sure. And it's nice because you used 8,000 protein ligand structures. We applied some filters of quality, resolution, B-factor, and if the ligand was complete, because that's another thing. If you have some parts of the ligands that are very flexible, you don't see it. So we don't use that ones in the training. But now you're doing like with new molecules, and you're having good success also. But of course, it's the first deep learning approach for that. So there's venues to improvement. So that's why also we want to share the database, because this database it was very hard to prepare. And we use it for semantic segmentation of ligand images to build new molecules. But you can use for in silico screening, you can use for many other applications. So it's we'll be there for all of you.
Yeah, a question here in the front.
Thank you. Super talks. Actually, two. I'll be really quick. One, you talked about using an exin mimetic peptide. If you get good activity, is there a tool that you could take to the clinic, or is there still a long development to do?
Yes, there is some phase 2 studies running right now for not infectious disease, but arthritis. So looks like it's good. The problem is a peptide. So what we are doing now, I had no time to show. We are doing some formulations to make the delivery better. So in our hands, what we are doing is formulation based on cyclodextrins, like the remdesivir. So then we are giving oral road. It's really nice. And also the effects on the inflammatory mediators, it's very, very potentiated. And also we are doing some formulations in non-lipid nanoparticles as well. So we are trying to improve that to try a phase 1, but there is some studies with the pure peptide for other diseases running out.
Great, thank you. And for Tiago, have you profiled MB905 against other viral families, just to see if it's an interesting tool for pandemic prep? I'm sure you have. But.
Yes, we did. We have some good results for other family of virus, but I can't tell right now.
Thank you, Charles. Srini has the last question.
I had the same question as Charlie, but continuation of that. Tiago, is MB905 being used clinically now for other diseases?
If you go to clinicaltrials.gov, you do find clinical trials, but it's not registered for any specific use. So It is used in humans, but as part of a clinical study.
Okay. So the reason why I was asking is you can directly enter to a phase 2A trial, proof of concept trial, without doing phase 1 right now.
I didn't hear well.
Can you enter into proof of concept trial without doing phase 1 with MB905?
We tried to connect some of this research, but we never heard back. To tell you the truth, one of the reasons why we choose MB905, because we want to push forward directly to a phase 2 clinical trial. But there was no appetite from the Brazilian regulatory agents for that. They are smiling because we also contact with DNDi, and they also did an in-kind evaluation of the material we had, and the general feeling was that we still need to do a phase 1 clinical trial, and there is reason indeed for that, now taking it seriously, because the patients with familial dysautonomia, they could have a very unique plasma exposure due to their primary condition. So in healthy volunteers, it's part of the package, and we need to do so. That's the plan. And this is one of the bottlenecks in Brazil, because we do see many networks of medical companies, and they're very comfortable doing phase 2, phase 3 clinical trial, or even phase 1 for compounds related to vaccines and bioproducts, that there's not the necessity of very specific timing for pharmacokinetics. So I think the bottleneck for small molecule development in Brazil is something very important to be overcome.
Thank you. I have a last question for Annette. So working on open science in drug discovery, do you have a perspective that different countries would have different landscapes for this kind of initiative? For instance, Brazil has history in breaking IPs for important drugs to treat the Brazilian population. So do you see a difference in ways countries treat open science in this field?
We had a lot of strategic discussions in a lot of different countries around this project. And I do think that there is a particular appetite for addressing IP, and especially restrictive IP, in areas where accessibility of compounds in the past has been difficult. And I do think it relates in particular to areas of neglected disease, or areas where funding isn't quite so available, like pandemic preparedness. So I do think it's going to be an ongoing discussion, and there's certainly some boundaries that are kind of breaking down.
Very nice. So in the interest of time, I'd like to thank all the presenters for the nice talks, and the audience. Thank you very much.
Now, okay. So this is the last session before the guys have seats, before the closing ceremony. I know people are excited to get the prizes, and oh, thank you. So, yeah, I thank the guys here for being seated, but I would like to thank everyone here, of course, for having me. So seeing friends, it's very, very nice. But first of all, thank Jadel, Luisa, and Luis for inviting me for this session, and to give me the responsibility as someone that it's not a communicator, or at least a communication expert. I'm here as a master of ceremony for the stars that are coming soon to talk about this very important issue for neglected diseases. But it's also an honor to be here to celebrate DNDi 20 years and LOLA 10 years, and also the science being done targeting the neglected diseases. So this is more important for us, and seeing the excellence of the Brazilian science, it's really, really impressive. Okay, so unfortunately for you, I'll have some slides to show before, just to introduce you this issue. Okay, sorry. Okay, we have now, for this session, we have Marina Verjavsky. Sorry, Marina, I trained a lot and then I did the mistake, of course. And Roxana Tabakman will be the real speakers for the section. And I'm feeling a bit adventurous now. Okay, I have no responsibility on communication, so I will give just my impression from pre-COVID and then post-COVID relationship of the scientists with the communicators and especially with the journalists. So it's my impression that we had in the past a very linear relation between the outcomes from the scientific projects being done by companies or even universities, that is used by journalists or communicators to deliver any kind of information to an audience. But it is very limited, I think, because we have projects, outcomes that were not exactly thinking on the population, not exactly talking with the people, especially in our case here, the neglected population, those that are affected by the neglected disease. And also, it was very specific coverage of any topic, scientific topic, that was kind of popular at a given time. And this is not solving any type of problems we have on education, and also to increase visibility of a given disease, of a given person suffering for this kind of diseases that we were discussing these days, which give me the impression that we had always a low engagement of all these actors, the scientists, journalists, and more especially the public. But then we are evolving in these topics. Science and communicators are working together, and the population is sometimes target for these projects. The people in the university, people in the laboratories, working together with journalists to have a strategy to fight, for example, misinformation, to increase education in a given topic, to increase visibility of those suffering from specific diseases. When we combine the efforts, what we are seeing is, including the citizen in the science that we are doing, we are trying to educate the population, not only those in endemic areas to prevent diseases, but also to educate people how to behave and how to talk with politics, for example. There are many other topics we can talk here. And again, to increase the visibility for those that need. And doing that, we are fighting the lack of funding, specifically for this disease, the policies and politicians that are not working in their best way to increase health conditions for poor countries like part of our country, to fight misinformation, and we also don't have any policies for this currently, or, well, any useful ones. And we don't have, at least at the past, we didn't have any way to de-neglect it, and I use this word, those that are suffering. Because we were talking to our peers and it was easy for us. But as I am now out of my comfort zone, we are all out of our comfort zone when we have to reach the society. And that's something that we put in mind in our projects. Well, the funding agencies now in Brazil are including in almost all call for proposals, any kind of approach for communicate the results, the outcomes, the projects, and trying to reach the communities beyond the university walls. And well, but we have examples. Luis, for example, is talking a lot during COVID, for example, for different kind of media, and we have many others doing the same. We have educating and trying to combat misinformation, but we also have DNDi, for example, just to give them my gift. That's the only thing I can give them for their birthday parties, doing the work on trying to increase visibility of those that are suffering from the disease. I'm repeating it, okay, but it's because of important. At least we will have this message. We will go to bed in pillow and see, okay, visibility, visibility. That's something we should do. That's something we should work and keep in mind. And I'm, of course, sorry for that, but I wouldn't miss the opportunity to show something that I'm very proud of, and it's a combination of work of a scientist with a journalist to show the situation or our showcase of how to not combat a very important disease. So it's you can contact me if you are interested, of course. Well, for today, the real stars, as I'm told you, I'm just the master of ceremony here. We are very ahead of time, so I'm trying to not use these credits, but okay, we still have some time to do it. We will start with Marina Warchowski. She's a researcher and science communicator. She will talk about misinformation. Then we will have Roxana Tabakman, a science journalist, will talk about visibility. And Marina holds a PhD degree in education, management, and diffusion of biomedical sciences. She got it from Federal University of Rio de Janeiro, my alma mater. She has worked on science communication projects with different universities and scientific institutes and scientific societies. Marina.
Professor, for the introduction. I'm also sad that Ayrton couldn't be here today. Oh no, I'm scared of that. So many people have fallen. No, it's okay. I try to stay still. So I was thrilled to see Ayrton's presentation today, but I'm also glad to share with you guys some of my latest paper that just came out last month at Vaccine Journal, along with my co-authors. I will start with a brief chat about why misinformation. Maybe some of you may be thinking, isn't that the same as fake news? And no, it's not. Because fake news is a term to describe fake news articles that come on newspapers and they're fabricated to pass along a false information. And misinformation is way bigger than that. That's not just fake news. And there's a clear fact of intention related to it, because fake news has the intention to spread misinformation. But we cannot always presume that intention. So nowadays, okay, here, okay, yeah. Nowadays, with in social media era that we are living at, we cannot just presume that everybody are sharing information and rumors and all, but not necessarily intentionally. Some people just fall into this net and they spread those misinformations. And we used to work analyzing fake news a lot back in the days, but nowadays we're focusing much more on the whole misinformation net on social media, so we can address that issue better. So, we analyzed some tweets on COVID-19 vaccines, but there wasn't just any tweets on vaccines. We chose a specific cohort of time period. And that was, I don't know how many of you are aware of that, but our previous president, no, our previous government opened a debate in order to decide if we wanted or not to vaccine our children against COVID-19. So we chose to cover the debate that was happening on that specific period of time. And until 1 month after there was already happening, children immunization was already happening. So that's just an overlook that we were able to collect over 1 million tweets and 4 million retweets from this period. And with we were able to see that were two different groups, one in favor. They were very clear that there was those two groups, one in favor and another one against vaccination, and they were very similar in size and retweets matching the tweets. That was already very sad to see, because it shows an increase, a massive increase of anti-vaccine talking on social media and also Twitter. So we analyzed that by computer models, but we also made a cohort, a small sample inside that big sample, made of influencers. So we analyzed 15,000, no, 1,500, it's 1,500, yeah, 1,500 influencers tweets, just the ones that got more than 500 retweets. That was our, and we also saw that those two groups were very similar in size, but the pro-vaccination group had more retweets, like almost double, but they were very similar in size. We analyzed that manually, and we observed 11 points, topics that were more prevalent in those contexts. And I will present them in order to most prevalent to least prevalent, all mixed together. If you want to read the paper, then you can see more context. But the most frequent topic was politics. It was obviously a quarrel, a political quarrel, mostly people complaining or praising ex-government. And as expected to be prevalent, children was the second theme. And in third place came themes about restrictive policies, just like freedom of health, freedom to travel without COVID certificates, and all kinds of freedom related to vaccination. And then disadvantages of vaccines. Then anti-vaccine people, and that was interesting to notice because some people praised, anti-vaccine people, and some others just complained about them. All of them were just like that. And in fifth place came mostly people complaining about misinformation sources. And that's, well, we were expecting that, but it's interesting to note that the pro-vaccine group attributed mostly the government as the main misinformation sources about vaccines. And in second place, they think that social media was a very powerful source of misinformation. And in contrast, the anti-vaccine group thought that traditional media was the main source of misinformation, in that case as fake news, because they were accusing them. Then in seventh place came international issues and examples as bad or good, but that debate. And just then came topics about advantage of vaccines, COVID risks, and science information. And that's very powerful because it's almost least position, and the group who most commented about science topics was the anti-vaccine group. And they were sharing like crazy some already debunked researches that were more aligned with their political view. And they also promoted a very intense dichotomic view of bad versus good science, and pointing out that vaccination-related science was bad because it was very related to pro-profit intents. So that was very sad to notice. And in least place, there were religion topic of discussion. We will see then that it was the least talked about, but it wasn't unimportant topic. So those two groups, they were very similar in size, but who are them? Who were them? The pro-vaccination group were composed from few scientists and science communications professionals, but mostly by unbriefed average people. And the anti-vaccine group was composed by journalists, neo-Pentecostal ministers, and politicians. So that shows low coordination from this group and high coordination. But other things shows that, but that is something that also. So the main findings were that we saw that anti-vaccine group was very proactive, campaigning against vaccine, and pro-vaccine group was merely reacting, and that was very worrisome to notice. Also, the fact that just the anti-vaccine group commented more about science, that's very dangerous. What can we do, right? First, I think that it is very clear that it is an information war going on social media, and we should all join this battle, because we need it, especially in Brazil. We believe that science institutes and centers should infiltrate better, especially social media networks, in order to fight this more properly. You all can also be proactive on this field. You can search for your communications advisors at your science centers to better learn what to do. But there are also some few tips online, like the debunking book. It's free, it's online, it's made by Lewandowski and Cook, that have been working on those issues for climate change for many years. And I will dare to say also, to finish, that you could try to partner up with some influencers on social media, because they are the real experts on this field. And there is a good case in Finland proving that they could be great strategic partners when properly briefed by health experts. So that's it. If you have any questions.
Thank you, Marina, for the great talk. And now it's time for Roxana Tabakman. I will just give a brief introduction about her background. She worked in different medias around the world, in Spain, Argentina, even in Brazil. And now she's in Medscape, am I right? And Noble also, and she founded the Rede Consciência, which is a network, it's a Brazilian network of science journalists and communicators.
Thank you very much. Well, first of all, Flavio, I share your thoughts, and so I will try to add something in the same line. And well, I would like to begin expressing my gratitude to be for the opportunity to stand in front of you today. And it's a real honor to be here. And of course, I will extend my appreciation to the organizers that brought us all together. Well, I'm a biologist. I'm from Argentina. I have been living in Brazil for the last 20 years, and I have been a science journalist for the last 30 years. The last 2 decades in health information, health-related topics. And in the last 5 years, I have been producing content for the Spanish and Portuguese edition of Medscape. Perhaps you don't know it because it's a global leader in medical information for physicians. At Medscape, I wrote several stories covering neglected diseases, and this is why I'm here today, I think. And there is a problem there, but as you know, there is each time more money for other illnesses and not for neglected illnesses. But also, I will say that even if I wrote many of all the subjects, in Brazil and in Latin America, the audience statistics, even among physicians, is very low when we publish information about neglected disease that when we compare to oncology, cardiology. So I will say I'm grateful to Medscape for the editorial decision not to abandon the coverage. Having said that, I will add a new goal for you after the many goals you have after these 2 days of meeting. And I ask you to remember when you promise eternal love for humanity and without limits. Well, today I will ask you to expand your limits and to work together in the shared purpose to giving the silent sickness a louder voice. If you prefer not to be in love and you are rational, science communication is an integral part of a scientist's responsibilities towards society. So just to remember, scientists can be fantastic, brilliant communicators. We have seen several examples these 2 days. You are training to present the documents for the grant, so you know how to do it. And in my experience in the coffee break, you're brilliant to explaining your research with clarity and purpose. So now I will give you 10 reasons to engage in science communication. Well, the first of all, of course, is to expand the knowledge reach, to go beyond your group. This was already talked today. To promote scientific literacy, that is also very important when we are talking about misinformation, but dispelling misconception and educating the public. Even the work professionals you work with, in Brazil we have this bad experience that health professionals have no science literacy. Of course, it facilitates information decision making. It has an obvious public health impact because each time you're talking about your research, you can create a new opportunity to address different aspects of the public health issues. You can raise awareness, encourage preventive measures, influence public opinions. Well, the fifth one is the one that I like the most, that's to share excitement and build trust in science, because I love science. And I have an experience this week. I was making an interview on Wolbachia mosquitoes, and the researchers were more excited talking when they went to school and they shared the love for science with the kids than for the experiment, because to share experiments on science is fantastic. Of course, to bridge a professional public gap, that means taking the knowledge out of the tower. Gain visibility and funding and enhancing career and network. All the journalists, we all have the experience that someone calls us, oh, you know, after the story you wrote, I have a new collaboration, I have a new funding possibility. It's not our merit, but of course it helped it. Is, well, publish or perish, I know that rule, but it's, perhaps you don't know that the studies that are shown in the media have an advantage citation, are more stated than the others. And of course, you don't have to forget the alternative metrics, that is the impact of research is not measured anymore only by citations, but also the reach and influence in the society. Of course, transparency, the people have the right to know what are you doing and how are you using their money. And perhaps if anyone read, can realize that I put 9 reasons and not 10, because I will leave the last one for the finish, so for you to stay, but also because I think it's the best reason. So, well, in summary, science communication serves a multifaceted purpose, ranging from disseminating knowledge and promoting literacy to fostering trust, accountability, public engagement in addressing social challenges. So let's assume that now you're motivated, but I need to tell the truth, it's not easy. There are many challenges, and so not to get you depressed, I put only 5 and not 10. But of course it will interfere with your work because it takes time, it takes energy. You have to overcome your own negative perceptions. I imagine many of you have negative feelings about journalists and communicators, sometimes you're right, I have to assume it. But sometimes because the information was ignored, because it was distorted, or was co-opted for political gain. So you have to face yourself to do it sometimes. Of course, complexity of scientific information that is not easy to spread in an accessible manner. You have to navigate biases and attitudes because people is not an empty cage. You have to communicate considering their biases and attitudes, and this is very, very difficult. And of course, and the last one is the rapidly changing media landscape that even is difficult for journalists to follow because they are changing all the time, the technologies, the platforms, and it's very essential to stay updated. And something I will add, just one more that is not exclusive, but is characteristic of neglected diseases, that they do not affect your audience usually, but it's crucial to reach the decision makers and advocates that yes, can be your audience. So this is an extra challenge. So I will ask you to think a little if you think that what is better, to talk about the diseases or to talk about the solutions? Because producing content, even is for other specialties, I'm not talking only in general audience, need to capture human interest. So what do you think is better? The mosquito is winning or Africa is winning the war against malaria? Well, I will take, people likes bad news. There are negative words in headlines increase consumption rates. There is a very interesting analysis that they analyzed 100,000 different variations of news stories, and they found that for the headline of average length, each additional negative word increased the click-through rate by 2.3%. But people like good news too. Negative news, news avoidance is high and is increasing. Why? Because the people is tired of bad news. A growing number of people is turning off the radio when the news came, scroll past the news on the social media, avoid certain topics in the news to be less depressing. And this also is a very fact-based data because it's the Reuters Institute Digital News Survey that is very, very good, that it surveyed 33,000 readers in 46 countries. The younger people under 35 are the ones that avoid bad news more. And this type of selective avoidance has doubled in the last 5 years in countries so different as Brazil and UK. They are 50% of the young people avoid bad news. So if the people like good news and people like bad news, well, probably everything is possible. You can present a neglected disease as a severe humanitarian tragedy, a public health problem, or an opportunity for innovation and economic development. The important is that to be accepted, the truth must fit people's frames. Framing is a technique of giving certain information priority over other information. Framing information can increase or decrease the way it is accepted by the audience, based on how well it aligns. So there is not a one-for-all solution. But thanks, it's good that it's not only these two possibilities. It's increasing a lot the solutions journalism, that the idea of solutions journalism that is very difficult for us to do. This is why there is very few examples. Is not marketing, it's not to show how fantastic it is to spread successful practices as a model. It can be done in another country, in something that can give hope, but with facts. This is functioning very well, and the people likes it a lot. Of course, this is a classical way to inform people, focusing on people or country for regional or local pride. Entertainment is increasing a lot. As you can see, we in Medscape use it for neglected disease. News gamification is a way to offer a more personalized news experience. It's a playful content that combines informative news with gamification elements, surveys, Q&A. The idea is to turn passive news consumption into an interactive experience. Of course, art is a way to communicate. This is a nice project of Fiocruz. And you can use even humor. This is an example I like a lot. This in the right is an article, it's in Spanish, but in English the title will be James Bond, Secret Agent with No Time to Die or Get Sick. And it's written after researchers analyzed in detail and published in Travel Medicine and Infectious Diseases all the health risks that are faced in the 47 countries that visited James Bond. I like this example. So let's imagine I have a success, you are already motivated, you have an idea in mind, what to do now? What non-communication can do? There is no one fits all rule, but there is one thing that is always important, that is timing. Perhaps you don't know, but the structure of DNA took one month to reach the press, but it was in the 50s. Today, science is spread as it happens. So, for example, if you are an author of a paper, it's expected that you give the embargoed information to the journalist to work with it, as the journalist can spread the news at the same time you do it. Well, you can also generate news, of course, ask help for communication specialists in your centers, because think that in the same way that you were employed because you know science, these people was employed, they know how to communicate, so assist them in the press release process, suggest idea, translate complex, make it, in a way, make a partnership with the science communication people of your institute. Make yourself available for interviews to comment other research workers. And if you like, you can try to develop effective communication skills. Not everyone needs to be a communicator, but if you want to, there are many media trainings you can do. There is the media trainer teacher said that it's very easy to communicate. It's just to answer how in simple terms and why in motivational terms. If you can do it, fantastic. You can be active in social media, of course. The best use to use social media is to deliver clear message via appropriate platforms tailored for diverse audiences. This is the gold standard. Of course, many of you are using social media probably to communicate, and you know that even if you do very little, you have to devote a lot of time and energy. You can write opinion pieces, and now I can recommend one to read, that is Professor Luis Carlos and Dr. Jadel published recently in the Conversation Brazilian edition, a very interesting article on neglected disease. And to write opinion pieces is very good because you can reach many people, but also it's a good practice because to write it, you have to have the key message in your head, reduce into a few points and remove all the technical distractions. So I recommend that anyone try to write at least an opinion piece on your subject. This is, of course, it's impossible to make a list because innovation is the best part. For example, the Minister of Science, Technology, Knowledge and Innovation of Chile, when she completed her doctorate, she found it too difficult to explain it. So she decided to convert her thesis in a video game with virtual reality. So innovation is the point. We at the Brazilian Network of Science Communication, we have been giving the last 3 years a course that is a shared course of the University of São Paulo and Federal de Paraná. And it's for postgraduate students only, and of odontology in both universities. It's a science communication is the course, but they are all the students are in the 20, 30 years old. Of course, they learn how to write because I teach how to write, but what they like the most, and they do it fantastically, is they explain their doctorate research in TikTok videos, WhatsApp messages, even memes with their own research. And I don't know how they manage, but they do it. So my recommendation is for people like me, be close to young people because they are extremely creative. Well, there are things that you cannot do, but I have not too much time. So the most important, if you decide to communicate by yourself, is to choose the target, considering that still the people read, but each time is less people reading. The audience, the people, young people prefer to listen and to communicate through social media. TikTok, Instagram, YouTube, all these are preferred for news consumption. So if they read the news there, only 75% prefer to read the news now. And in any case, the first rule is do not use jargon, even to communicate with your colleagues, because they are colleagues, but they have been, today everything is interdisciplinary and very speciality. So to communicate is each time difficult if you use your own jargon. There is one point I didn't forget to talk today is if you produce content, of course you have to take care with the words, but you have to take extra care with the images. You know that the cell phone is something very small, but can can do a great damage. And the choice of images is what shapes the narrative. So especially on health and disease, especially in low and middle income countries, the images need to be carefully chosen because they elicit emotional reactions. So what is right is to use images relevant to the topic and with respect for individuals. What is wrong is what people use in the past, but they are still using. That is to generate pity, and because this is a perspective that produces harm. Well, and if you do not know how to begin, well, of course, personal relationship with journalists is always welcome, and I strongly suggest it. But as you will not find us at the bar, I suggest you to include your name in Source Bank. For example, in Brazil, the Body Agency is a press support service in which we, the journalists, look for sources. The Science Media Center is a British institution that also have an important database of researchers. So in summary, it's not easy. I will not say that, but it's to all of us, from scientists, journalists, policymakers, to promote accurate, reliable, and accessible scientific information to the public. I highly encourage everyone to make the effort, as I'm confident you will find and develop the tools necessary to do it. And now, well, I will, for those that are still asking why you spent 20 minutes with me, I will finish my presentation and I give the last reason why to communicate. I'm the author of several books, including Health in the Media, Journalism for Physicians, Medicine for Journalists, that was published in Portuguese and Spanish. And the goals of this book was to bring together physicians and communicators in a mutually beneficial partnership to better inform society. However, today I find more relevant my latest book, that is Biovigilados. It can be translated to Biomonitored. That it was published by the Penguin Random House in Spanish. And it's about a respiratory virus that produces flu-like symptoms but can be deadly. It's priced worldwide from an unknown source in Asia, but it's not what you are thinking. It is a science fiction book that has more science than fiction, because I'm a journalist. And it was based in extensive journalist research, and the ones that were yesterday, remember that Professor Elena Nader took us, remember that we were waiting for the pandemic many years ago. Well, this is what I have read also. And just the characters and the plot were created by my imagination, but it's not a pandemic book because it was published in 2017. So it was written a few years before the corona. So as a medical thriller author, I will ask you a favor, and this is my take-home message. And it's not, I'm a science fiction writer whose vision unfortunately came true. And so please don't remember that many of major calamities start with the scientists not being heard. So please give the silent signers a louder voice. Thank you.
I invite Marina to have a seat. And of course, I open for questions. And also, if you want, you can share your experience on science communication.
It's interesting. Is it interesting the tweets you analyzed on Bolsonaro narrative? but it's more wider than that now, right? So we have a kind of science movement, such as arguing on the origins of the coronavirus and how it is supported by different groups. So I would like some on this more broad narrative on anti-science movement and how they are defended, including social media. And I'd like also to share our experience. For Fiocruz, we have an agency for news and share our results with them once they are published. And it can become something very short as a note in a newspaper or primetime television interview. And in 2 times, some of our work on reproposing outreach, clinical research that contact us to do clinical studies on the compounds we were analyzing. And this became the source of our new network. So this was very important for us.
Well, yes, it is a big problem, and it's go way beyond just the political quarrel related to Bolsonaro. But well, we must focus our research on a specific point, and in order also to compare with other researchers that look at other situations and how people react on those situations online. So I'm not sure what else I could add, but it is very interesting. Fiocruz has a big communication, including an institute that teaches science communication. It's not the case for most science centers in Brazil. We see a real lack of real effective communications in most of Brazilian science centers. And well, you have something to...
No, the problem I see more, I work for a website for physicians, and they were all Bolsonaro, or many of them were Bolsonaro followers, and many, when we have to, many stories during the pandemic, we have to take off the commentaries because they were against science. So it's difficult to explain that.
Hello. Yeah, I had a question. Do you think the science communication should become a part of a syllabus for undergraduate students or even graduate students? So you can start at that level, so where they can start instilling in themselves how to communicate their science across different forums. What are your thoughts? I'm talking about making science communication as a part of curriculum for undergraduate students, start training them at that level to communicate science publicly. So then it becomes more visible. Any thoughts on that?
Yeah. Did you understand me?
If science communication should be part of the curriculum.
Yes. Well, what is talked now is, at least when you begin a research, you should begin to think about the communication. This was clear after related to vaccines. This was clear because till Corona, we have no problem. The people go to have a vaccine and didn't ask the name of the vaccine, when was created, what it had, because they have no choice. They have only one vaccine, and they go and receive that vaccine. With Corona, we have several vaccines, so the people begin to think on that. And no one had thought before that this will be a problem, because never in the history we have several vaccines to choose, or not to choose, or someone to choose for us. So this is an example of how when they begin to think on different vaccines, they should have begin to think in a communication for that. And of course, today the division between communicators and not communicators do not exist anymore. We are all communicators. We have all social media. So yes, it should be the school curriculum, because how to read social media and how to spread information on social media should begin during from the infancy. The kids should learn. But of course, in the university, I know that, for example, some scholarship for students, summer programs. I know, for example, the summer program of the Institute of Weizmann in Israel, and they include science communication as part of the curriculum in a 2 months period. So I think it's worldwide the decision.
Well, thank you very much for both of your presentations. We really appreciate that. I think that COVID-19 has opened everyone's eyes to the dangers of misinformation. Misinformation can kill. Misinformation or fake news or whatever you define that is like a virus. You know, it really can kill. It's really danger to our democracy. We are talking about science, but we saw this same example in politics. Argentina is facing the same problem right now. They have an election next Sunday. So, I mean, we need to understand how this propaganda in this 21st century works. As scientists, we need to reinvent ourselves, I think. We are not going to have how can I say, state politics. Okay, of course we need to start by educating our children, but it's a very complex problem, and of course the solution is not going to be very simple. So I'm not asking a question, I'm just making comments because I had the opportunity, you know, during the pandemic to fight against this, I mean, very well organized ecosystem, as you mentioned. They are very well organized, they are financed, there are many, I mean, interests. There are, you know, politics, religion, financial interests, and we were not prepared. So, I mean, how can we get prepared, you know, because we are going to face, you know, new problems, new epidemics, new complex crises in the future. So we really need to get prepared, and the solution is really not easy. I'm not asking a question, just making this comment if you want to say something, but it's really tough.
It's not, of course, there is no one solution, because I think that it's of the Nobel Prize, the person that know how to resolve it. But of course, there is a lot of politics and economical interest, but there is something that we have to, as communicators, to make a mea culpa, because, for example, we never show uncertainty. When we did science communication, we showed the good things, the discoveries, and we arrived to the moon, but we didn't show all the experiments that did not give good solutions. We did not show how we take decisions without knowing all the information we need. And actually, physicians never do that. They give you a pill and say that it will cure you, and they do not say that it will be a 60% possibilities that you will be cured and 40 that you will not. So not to show the scientific process is something that do not help to scientific literacy. And this is something that science journalism should change. But solutions?
I don't know about all the solutions, obviously, otherwise it wouldn't be a problem anymore. But we must do something, because what we've been doing is nothing, and it's just reacting poorly for what they've been doing. So it's clear that something must.
One thing that is easier, easier to say than to do is all the recommendations is not overreact and try to have empathy with the people and give information.
That's
is
one point.
The theory, but I cannot do it.
Yeah, I forgot to mention about, but it's important because in the debunking book, it's a very important topic. It's not to just judge people for their misconceptions or even to fall into into those misinformation nets. Because some people just have reasons for that. They're afraid. There are several reasons that may lead people to follow on those misinformation nets. So we must be careful not to judge them and put them all in the same bag, like they are all badly intentioned. And the second one is to not forget to give the right information. We've been failing to do that on social media till now, but we can still.
Yes, there are some techniques. For example, you have to debunk an information. You should talk first about the good information, then comment the bad information, and then talk again the good information, like a sandwich. It's a recommendation, but have no evidence that it functions or not.
Yeah, they have some. Yeah, those people have been studying misinformation from climate change for a long time, and they have some insights that it works better, not to judge people on social media. So I think it's an interesting book. It's free, it's online, so everybody can just take a look and have some insights.
Okay. So thank you. Any other question? Oh,
me.
Renan.
Hi, I'm Renan. I'm a PhD student here at Secamed. I would like to take advantage of this open mic and share some ideas, provocations, and so on. And I would also like to hear your take on what I'll say. So to begin with, I don't think that science communication should be on the syllabus for undergraduate students, because a lot of them will not engage further in science after university. So a lot of them will go to sales, will go to other areas where they will not use science in their everyday. But I do think it should be a mandatory part of the graduate curriculum. Because when you engage in graduate studies, you are almost always sure that you will in some way advance in your career with science. That being said, I also see a lot of professors and scientists in general who say they do make science communication, but when you check it, it's just they are posting their papers on the link of the papers online, and that's it. I think we should take a lot of care with the language we're using, the media. So the same video that will work on Instagram will not necessarily work on TikTok, for example. And it's not just the duration, but how you talk to the public. I think this is the kind of thing that we should always be careful with, because it will impact on how it will be perceived by the public. And I think that this type of thing should be always part of every symposium like this, because even if we make today a mandatory part for graduate studies, there is already a lot of generations that are already working that didn't have it. So I think it's a workshop like this, symposiums. I think it's always good to have this type of debate, because this way we can at least try to catch up with this type of thing. So one last thing I would like to share is something I don't see a lot in Brazilian science communication is talk about the Brazilian scientists. So if we take the Brazilian public perception of science published by the SBPC, we see that Brazilians do like science. They always say that the majority of Brazilians like science. But when asked to name one scientist, 93% of them can't name one scientist. So this is started working on science communication. I'm with the t-shirt of Via Saber, my group. So our goal is to invite scientists to speak to the public in general streetways. So we started on Avenida Paulista in São Paulo, but we have already went to 11 cities in Brazil. And it's always nice to see how the population perceives scientists and how they're always glad to meet a scientist, to see that science can be made here. When we were getting sources for this project, we talked to a professor that talks with children about where science is made, how it's made. And one common answer they get when he asks, like, where science is made is in the US. So people don't know how science is made here. People don't know Brazilian scientists. So I think we need to talk more about the history of our science, the present of our scientists also. So to engage these, I have this group, Via Saber, that gets these professors in streetways, and also my book, Super-Heróis da Ciência, Heroes, Super Sciences, that talks about 52 Brazilian scientists and how they impact the Brazilian science and international science that we usually do not talk about. So sorry for speaking that much. I would like your take on what I've said.
I just wanted to comment something that for scientists to be in the media is a very new thing in some countries. And for example, if I call Harvard University and I want to make an interview, I'm sure in 24 hours I will have the interview. Brazil is good. In Argentina, I can stay 2-3 weeks waiting for the researcher to answer. So this is true, it's changing, it was worse. And also you have to think that the media is much quicker and cheaper to translate an article from the United States, from the New York Times, than to put a team of science journalists to work on a story. So this is another reason there is much more information that is being translated. It's cheaper.
Yeah, I loved your position, and as a press advisor, I worked as a press advisor for many years, and we faced a really difficult time to trying to put some research into newspapers in Brazil, because we facing newspapers, science editorials, and they are almost dying here, and it's very difficult and contributes to that problem a lot.
And also you have to think that there is a globally crisis in the media, because now information is free. But it's not free. You have to pay the journalists. So what happened, all the journalists, the big media is closing their, it's an economical crisis that impacting the quality of the product.
Okay, so for now we are finishing. I just want to share my solidarity with Marina, but I cannot say how depressing was reading all those tweets.
Yes.
Oh.
It was very much
terrible.
depressing. Yeah, really
And
sad.
thank you. We are right on time for the closing ceremony and for the prizes, of course. Thank you again, Marina and Roxana, and you all.
Okay. Is Jadel around here? Here. And Luisa? Okay. And the books? Okay. Thanks. So guys, we are coming to the end of this meeting. I mean, we had 2 great days, 2 fantastic days here, and I have a lot of people to thank. So first of all, of course, thanks to all of you for being here, attending this meeting, this LOLA 10 years celebration. Thanks a lot to all of the speakers. Thanks a lot to all of the colleagues that, I mean, worked as chairs of all of our sessions. Thanks to the Institute of Geosciences for this facility. I can't forget this. So I have a list of things and people to thank. Thanks to the DNDi, of course, for this partnership during these 10 years. Thanks to the DNDi team, especially Charlie, Peter, Sergio, Jadel, Luisa, Marcela, Marina, Renata, Raquel, Laura, I mean, everyone from DNDi involved in the organization of this meeting. Sorry if I forgot someone. Jean, it's over there. So thanks also to our colleagues from USP, Glaucius, Adriano, Leonardo, Carolina, and everybody else that helped organizing this excellent symposium. I would like to congratulate the DNDi again for the anniversary of 20 years of, I mean, an inspiring, beautiful, fantastic history. So congratulations. I'm looking forward for the next years. I would like also to wish the best of luck to our colleague, Professor Musumi. I don't know whether or not she's still around. Good luck with the Loki Consortium, with his, hello, with his, the Lead Optimization Consortium in India. So thanks also to FAPESP, and start for the financial support. And that's it. I think now we have, Jadel has something to say, and we have the posters, okay, prize. Thank you, guys.
Very, very short, really. I just want to thank you, everyone. I hope you enjoyed these 2 days. I certainly did it. Our DNDi team, Luis, and everyone involved on the organizing and the scientific committees put a lot of thought and energy to make this happen. I'm really happy we are coming to an end. everything went well and smooth, and I hope you enjoyed the discussion around how to advance drug discovery in neglected areas. It's great to be talking about the challenges and the problems we have to face and how to overcome them, but of course it's great to also celebrate the great science we've seen globally, locally. It's great, and to highlight how important it is to collaborate and to coordinate our efforts, and of course to celebrate the 10th anniversary of LOLA. Once again, I would like to thank my DNDi colleagues, Luis, thanks a lot, everyone in the organization, committee and scientific, FAPESP for the funding, Unicamp for hosting us, all the chairs, all the speakers, you made it possible, and of course, all of you who attended here and online. So once again, thank you very much and see you soon.
Okay, I'm using this mic because I think this is the best one. Okay, so first I want to also thank everyone for coming. It was a huge effort from everyone, but it's special thanks to Laura and Raquel that made everything possible, also with the agency, Hilda and Flavia. And also my two bosses, my former boss, Luis, and my boss now, Jadel. Anyway, so for the prices, let's finish. I have written down here. First one, the first prize goes to Tanidis. I know that she's there. Come here, Tanidis, with the poster. Yes, I'll do it. So her poster title is Synthesis and Biological Evaluation of Glycogen Synthase Kinase 3, GSK3, Inhibitors Aiming at a New Treatment for Leishmaniasis. So the price is let me read the title, I forgot. Fundamentos de Química Farmacêutica Medicinal. It's our Brazilian book of MedChem written by, of course, Dr. Eliezer Barreiro. And so we have first the newest edition here given by Professor Lydia, where she is, I don't know if she already left, so she had to leave soon. So anyway, so this is for you. So we have two other, like the runner-up prizes, so we have two of them, and I'm gonna read both of them together. So the first one is Igor de Andrade Santos with the new synthetic palladium 2 amino advantine complexes strongly impair SARS-CoV-2 infection. Come here, Igor. And the other one is Virlanio. Is he here? I don't think he is, but he's from Unicamp, so we can give him later. So his title is Red Propolis Purification by Flash Chromatography, Preparative TLC, yields a new anti-Leishmania brasiliensis fraction. Congratulations, Virlanio e Igor. So we'll give Virlanio, if we remember. Maybe no, we will. Anyway, so this is, guys, this is the end. Thank you everyone for coming. We have a very short reception at Unicamp now with some beer and caipirinhas and, I don't know, pastel or something like that. So we welcome everyone to go there. Okay, oh my God, okay, let's go.